Let me do that real quick. And this is my friend. He has consultancy and he's trying to stand up some AI stuff and win some big contracts. And he needed some help, so I am helping him with this. It's all open source though, um, and yeah, and we intend to keep it that way. So, and we're just gonna make money on consulting. So, okay, so let's see. So actually, this is a good slide for everyone. Uh, so cell typing, which means okay, let's back up. In one thing that has become pretty prevalent in the world of biology in the last 5 years or so is what's called single cell transcriptomics, which means you can sequence the RNA, not the DNA, but the RNA that is being expressed in single cells at a time. So so first of all, DNA is sort of like the copy of the software on your hard drive, and the RNA is like the running software in memory, is a good way to think about it. And it's not a perfect analogy, and it's like maybe hardware analogies are appropriate as well and whatever. But the RNA is just basically what's being done now in the cell, as opposed to all the things that all the proteins that the cell can express. So there are various technologies and whatever, but they all basically boil down to for a given cell, you have a matrix of like 50,000 or so genes, and just count how many of each of these genes you can detect in that one cell. And these matrices are generally very sparse, so about 1% of in any given cell, about 1% of the 1 to 2% of the genes are being expressed at the time of the transcription. And this process is also like highly dependent on the experimental setup. So it's very subject to what's called batch effects, meaning the guy who run it, spent a little bit longer during the sequencing process, or they used a different brand of reagent, or just basically there's a lot of randomness in the collection process. So these batch effects cause a lot of headaches. And so that models that are able to sort of like account for batch effects, that are able to sort of extract the gene expression information, and then figure out what, for this exact cell, what type of cell is it? And that could be as coarse as, is this a neuron or a muscle cell? Or that could be like, given this like super specific, like I know this is a neuron because it's from a chunk of the brain, but I don't know what type of neuron. So, or maybe it's either one of, so basically typing the cell, and this is like upstream of a ton of really important biological types of experiments and diagnoses and whatever. So that basically we've kind of identified this as a really core problem that has a lot of academic there's like tons of research that happened even a couple years ago about this problem and how to solve it. And people got to the point where they're kind of happy with the solutions from an academic standpoint. And then it really hasn't made its way all the way into practice. And so it's like pretty tough to use these models. Not that, like, for you, it probably wouldn't be tough, but for a biologist, it's kind of tough. And so, so we're kind of focused on, let's take this up, wrap it up into something that is really easy for people to use. And so that's the point of this. And I'll give a little more detail on that. Okay. So, so like I said, there's like the state of the art kind of is pretty either coarse grain or really specific to specific types of cells. So like this on the right hand side is like a hierarchy. This is an ontology. There's like a database out there that you can use that is basically the data sets that we use are normalized to this. So like there's like so the problem that you have is if you make if your data is labeled as like lymphocytes or like innate, and then you predict CD16 positive, like, is that an incorrect prediction or how correct is it? Is it's not a simple classification problem. And then of course these can be wrong as well. So, but maybe if it's like a different kind of mature natural killer cell, like how wrong is that really? If so, that should be like seen as different than predicting it as a, I don't know, like a muscle cell or something. Um, so, okay, so cell typing, right? Okay, you guys already know this. This is a model. It's a transformer-based model. It was trained. So, like, without getting into too much detail about how it works, um, like the data sets are not. So unlike gene genetic sequences, so nucleotides, like, you have the four nucleotides and they go in these sequences and they turn into these double helixes and blah blah blah. This is not that kind of data. This is for one cell, I have a matrix of 50,000 possible genes, and what's the number of times I saw that gene? A gene is like, like a string of maybe anywhere from a couple thousand to a couple million, I think. No, probably a couple 100,000 genes. Or sorry, nucleotides. They all string together, A, T, C, G, in different orders, and that defines what a gene is. So anyway, you get these counts. The count, obviously, because it's just a matrix, it doesn't really matter what order the genes are in. So you don't have quite a one-to-one mapping of language modeling to uh, of this type of data. So the authors did some stuff so that they could express the data in a something like an autoregressive model. And it's a little unclear to me why they really want to do that instead of doing a more of like a encoder only model. But it does perform well. So, but like I think in my mind there's a lot of controversy around, is it because of the other stuff you did and you should have used a BERT type model? So anyway, for what it is like a autoregressive, more or less style of model, similar to GPT. And it was designed explicitly to remove these batch effects. And this is going to matter in a minute. Um, and it does pretty well. But you can see these data sets. If you look at, well, you can't see it in this column, but like the data sets are uh, um, the data sets are these small specific types of cells. So they're not doing like, give me like of all the cells, 700 or so types. And maybe if you look at, maybe so even a big subset of that, like they don't do very well with that. Okay, so I know this is like a whirlwind tour. I don't want to dwell on anything, but I do want to make sure people kind of get the gist of this. Is there any questions to start with? Okay, cool. So let's talk about what I did. So one other background piece. There's this other model, and this is, I think, the reason why I wanted to share this was because I think this is a super duper cool use of embeddings, and it's a little bit counterintuitive, or that it works so well until, or maybe some of you guys might be like, yeah, obviously, but I think it's awesome that it works super well. It's actually surprisingly great, but um, really simple to implement, super efficient. And so I wanted to share this with you guys and like sort of the exploration that I've done with this and a small model that we've built on top of it and where we're going with it. So, so this gene PT is basically built by taking first of all, for every gene in my 50,000 or so genes, I have what's like a description of that gene. You start with this thing called the NCBI gene database. And for every one of those, I can just get a description. This gene, you know, um, this gene, like I could even maybe zoom in. No, I can't zoom in, but like, this gene is used for creating this kinds of proteins and like, is this, like whatever, right? It talks about what the scientific literature, some small summary of what the scientific literature says about this gene. And then you take that, you pump it into this, I didn't do this, sort of like GPT, the OpenAI embeddings. and you get out an embedding, right? And so that's like a vector that is, between 1024 and 3078 dimensions. These are, by the way, Matryoshka embeddings, so you can chop off the higher dimensions and they still work well. And so you take that embedding and then you look at, for the cell, what is the gene expression for each one of those genes, and then you multiply the embedding by that scalar. Like, so if I have like, in this particular cell, I have 6 of this type of gene and 7 of that, then you take the 6, multiply it by the embedding of that particular gene. So, all your embedding weights will multiply by 6 in that case. And then another gene, I saw like 100 of this one, so I'm going to multiply that one by 100 and whatever. So so you added all this up, you normalize to 1, and you end up with this embedding of that cell based on its gene expression. Okay, I want everyone to understand that, because that's if I think it's obvious if I explained it right, but maybe not a very good explanation. So I want to make sure people are on the same page here. Any questions? I'm not watching the chat, so in fact, I can watch the chat. Oops. Okay, it's just about transcription. Okay, anyone have questions? I want to make sure everyone's clear here. Okay.
Sorry, where do you get the gene expression level from?
Okay, that's from, so I haven't covered that, so that's why you don't know that. The answer to that is there is a database there are 2 databases. A database that was compiled by the Chan Zuckerberg Foundation called CellX Gene V2. And it's basically, they collected 100 million cells, so like 1,000 files that sum up to about 100 million samples or cells of this type of expression data. And it's publicly available. It's probably referenced in one of the links here. I'll share the slides when I'm done, but like you can like anyone can download this. So we also this company withMIRAMICS also compiles these data sets, and that we have access to larger and non-publicly available data, but but anyone can download the data that I'm talking about. Does that make sense? And then there's another data set that is a subset of that is like very carefully curated to be good for a benchmark on single cell, or actually it's not a subset, it's removed from that data set, and it's a benchmark on your performance of whatever algorithms, but you're supposed to use it not for training your models, but actually benchmarking your models. That's called Tabula Sapiens. And both of those are Chan Zuckerberg Foundation. If you don't see a link here and you want to see if you want to know how to download that, I can share it with you. Does that answer your question?
Yeah, thank you.
Great. Okay. Sorry. Okay.
Yeah.
Hopefully yeah, go ahead, please.
Question. So, do you have an intuitive way to think about the calculation you're showing on the slide right now? Like,
Yeah.
is it projecting from one place to another?
Yeah. So I think that so each gene is represented by a direction, right? Which is and this is in a high dimensional space. So that 3078 dimensions. So each gene will be somewhat, but not completely orthogonal to the other one. There'll be like a general direction that I'm describing a gene, right? All of them will be in that direction, but then like each one will be slightly different direction. And when you add them all up, you're kind of getting the average, and then normalize to 1, you're kind of getting the average expression of all these different genes. But the interesting thing about that is that because it's a text embedding, then like, let's say the information about the embedding is like, oh, like experiments show that this is related to cancer, right? Or something like that. Then there'll be like some bit of cancer direction in there too, right? And whatever, like drug interactions, etc. So anything that's mentioned in there, so that you're getting like sort of a, there is a sense in which the direction that the cell is pointing will be like sort of the aggregate of the scientific literature about all the genes for this particular cell. Does that make sense? I mean, it's a little bit abstract by its nature, but that's the way I think about it.
That makes sense. And so when you multiply it by your the matrix that's kind of green and blue,
Yeah.
that's all that cell the gene information you had on the previous slide, right? Like it was a big block of
Yes.
for each one.
Exactly.
And
Yeah.
then what you get in the output is for that specific cell, all that specific gene information, but for that cell.
Yeah. That's right. Exactly.
Okay, cool. Thank you.
Yeah. And it actually turns out that this technique, in some cases exceeds the performance of other, like the SCGPT, for example. Which is kind of incredible because the SCGPT transformer was trained on like actual genetic information, whereas this is just like, I'm gonna, so that like, presumably there's like a lot of relationship information between different genes and whatever. Whereas in this case, we're just saying, like, oh, we have these different, embeddings, and we're just going to create a new embedding space, and then like look at how well we do. So like, and you can see this UMAP over here, and I have a few more, I think, UMAPs in this, but you can, or no, it's not in here. It's in my notebooks, which I want to make sure I get to. But you can see like, okay, like your clustering is kind of capturing, the colors are the cell types. So like, it is kind of capturing different cell types in different, clusters in UMAP. So that's promising. So, and then like, since this is like a, latent space event, we have to have a graph of performance, and we can see that, like, I think my nutshell is that sometimes SCG in some of these data sets, right, SCGPT does better. In some data sets, GMPT does better. And there's like, I'm not going to go into the difference between these two. But and then some, like this is the exact model. So in this Bones one, and then in some data sets, the ensemble between the three of these does better. But one thing that is important is that even when it's not the top, the ensemble is always pretty close to the top, right? So like, so that what this tells us is that the ensemble probably will do better. And like, maybe also interestingly, sometimes, because like sometimes SCGBT does well, sometimes, and does pretty significantly better than this in accuracy and precision recall F1. And then sometimes GenePT does the best, and it does pretty significantly better than SCGPT. So they're capturing different information, right? So an ensemble is a great a great approach because you can kind of hopefully get close to the best of both worlds. So yeah. Okay, so let's talk about, I'm going to explain this plot a little bit, then I'm going to dive into some notebooks because this is AI in action, not a paper club or whatever. So this so what we did was we added to those descriptions. So remember these descriptions here, because in the original paper, they use these just basically the descriptions from this NCBI gene database. What we did was we got GPT-4 to give me more information about tissue type, drug associations, and pathways to improve the embedding, right? So we embedded we generated text on each gene using a prompt. We did that over all 50,000 or whatever genes. And then we took those and we embedded the full description. So we came up with our own. And with this is not super well labeled, but for different, so we have this smaller test set, you have like, I think 20 donors of cells, and then, or so, and then just leaving, I picked 3 that I thought would be good for test sets. And then what we did was just leave one out. So train on all the donors except this one, and then test on the last one, and then do the same for each donor, right? And you can see that like um I think that there's 2 things that I think are noteworthy here. Is one is that um like adding, so this red one is the original embedding basically, and then these guys are improved embedding. So it does actually seem to help to add more information about the cell, and just like these are like progressively adding more information to the prompt, and it does continue to get better, right? I mean, not in this case maybe, but in many cases. And the other thing to note is like this is SCGPT, and this does pretty significantly better in some cases, right? So which we knew. In this case, it doesn't. And then if you look at the weighted, meaning for by the number of cells, then actually in this case for this donor, we do this done is pretty significantly better than SCGPT, and even in the unweighted case, it does as well a little bit. Okay, so that's like sort of, so I want to just talk a little bit about how I did this. So I'm going to switch to, let's see, I have a couple workbooks, not this one, I want this one. Okay, so one thing, the other thing is, since this is AI in action, I just want to talk about, I think I have this workflow. I actually am curious to hear anyone else using notebooks in Cursor or another another, sort of VS Code base, or like, what is your, I'm curious to hear about other people's workflows with notebooks. I have a love-hate relationship with them. I love that they're so flexible, and I especially love that I can see, I don't have to like render, I can like sort of flip back and forth between cells. I can render. So for exploratory stuff, they're awesome. They're amazing. Even for some pipeline stuff, they're amazing because you can see sort of plotted output for your pipeline. But they're also like total garbage for rigorous software development because they basically have global state, right? And so that you're programming like basically that has like no concept of isolation from other functions, right? So like, what do you guys, I want to just pause and ask, is anyone else using notebooks? And if so, like, what do you do? Any takers?
Is my okay, my mic's working.
Yeah,
Yeah,
yeah.
Jeremy Howard has like a whole workflow for working with Python notebooks where they do like production level stuff. I have not quite adapted it yet, but I actually believe they produce some good software over there. It's at Fast
Oh,
AI.
yeah, yeah,
Use
yeah,
MB Dev. If that's something you're interested in, you want to look into, I really respect what they do over there. So I
yeah.
think
I mean,
that's
I've
a cool way
I've
to
done,
develop, but I don't personally use it.
yeah. I mean, I definitely have done the I've done the Fast AI course, and um at least one of them, and like it. But does he have like a specifically like, here's how I do notebooks in production kind of thing?
Yeah, MB Dev, I think is like what they build on top
Oh,
of. And if you go look at like
okay.
Fast HTML, it's built with it.
Yeah.
Flash, there's a bunch of
Okay. Yeah.
tools that they build.
Okay. Definitely gonna check that out. Thank you.
Yeah, yeah, no problem.
Um, and I know that I know Slono hates them.
I can't get used to them, bro. If I'm being honest, like it's just yeah, making that switch is tough, I think. But
Yeah.
I think it's a cool way to develop if you've adapted it. And in MBDev, they have some cool primitives where like you kind of just like develop in the notebook and then all your docs are auto-generated. All of your like the READMEs auto-generated. Like a bunch of I think even the doc strings get auto-generated. Like there's a bunch of cool primitives that they have in there to make it
Okay.
easy to go from notebook to like actual production stuff.
Nice.
But I haven't done enough of a deep dive to like really. say like, oh yeah, I'm ready to switch. But I
Yep.
respect it, and I think it just shows that there's another way it can be done.
Yeah, yeah. Okay, great. I love Jeremy Howard and all this stuff, so definitely gonna check that out. You want to say something, Mino?
Yeah, I mean, yeah, I hate them. But I worked a lot with like material scientists and like chemists and mechanical engineers that would produce these like absolutely horrid Python notebooks and then say like, can you
Yeah.
push this to prod? And be like,
No.
I one of the techniques that I developed is just like have a Python file on the right that you import into the notebook and then do everything that's like stable enough, move it over to the package and then
Yeah.
reuse it in the notebook.
Yeah.
And then the other one that I would often use is replace the notebook with a script that's the same as the notebook, right? Like something that is not meant to be functions or a program
Yeah.
that you reuse, but add decorators to cache the expensive to compute data
Mm.
so that the first time you run it,
Interesting.
it will do that, and then you can always run the whole script, right? So you don't, so that you start with the fresh Python, don't have like objects left over from like 14 cells ago that are kind of gone, and that would develop on that. So it looks like kind of like a notebook, but it runs as fast as a notebook to get to the stuff you're working on, but it is actually reproducible. Like those were
How,
two techniques that I used.
how did you sorry, where does the decorator save the state? Like onto disk or
Yeah, just like dot
okay.
state as JSON or something like
Okay,
that. I'm
I
sure
see.
there's like
Okay. Yeah.
more elaborate.
Okay, but
Um,
it's saving it to persistent storage so that when you redo then but the other thing, so maybe you have a thought about this too. The other thing that I really like is the ability to have rich output in between cells, right? Because then I can see, like, even if it's like at work, I often we use Databricks, and some of the pipelines are written with notebooks. And the nice thing about that is that you can be like, okay, here's the output of this intermediate step, and here's a plot or a table or something, so that when I go back and review because of a failure or whatever, I can just look at the plot and see the intermediate state.
there's this lightweight Python thingy where it's a Python file, but you can use like ampersand, ampersand, or like %%. There's like a syntax that eludes me right now, which when you open it in VS Code, for example, looks just like a notebook. And I think it can render graph outputs if you have a matplotlib thing in there.
Okay.
But it's still like a .py file, and you don't have this like IPyNB JSON
Yeah.
monstrosity like lying
Yeah.
around.
Right.
So that I found pretty useful. And then this was quite a long time ago, but I remember that Airbnb had like a notebook publishing system, which would make some really nice renderings of Python scripts. Basically, that's what I used. That's what I had the data team that I was leading kind of use. I think it's called Airbnb Knowledge Repo.
Okay.
I
Nice.
I think it was deprecated back then, but it looks like it's picking back up again. So I mean, it's 2 years old, but this allowed you to like just publish a Python script as a rich kind of
Interesting. Okay.
output.
Yeah. Okay, cool. All right, great. So that was a really helpful sidebar for me. Hopefully this is being recording, so hopefully I can go review anything I forgot. So and actually, just as a side note, that workflow that you mentioned, where you sort of have like these side, like things that are like useful, you kind of like move into actual code, and I have done that very much. So like so like this inference for that GenePT for those like the GenePT cells, I've kind of codified into this like this set of libraries. So here's like how do you do the inference? This is embeddings. This is all open source. I'll share the link. And but like so you can see that there's different and like a bunch of utils, right? So I call these from my notebooks, and as I develop them, like, and they become more like, I'm like, oh yeah, this is a common thing, or people would benefit from this, and I add them there. Um, so okay, good. So let's see. So where do we start? The this so I did this, I think this is the right notebook. So I did this EDA. Oh no, this is not it. I think this one is the one I want. So this is, as I mentioned, this is this Tabula Sapiens data set. This is like, so CZ is Chan Zuckerberg Institute. You can, this is a link. I think the easiest, if anyone's interested in like following up on this more, I'll just post a link to this repo that I'm presenting from, and then you can just like find all this information. So this is like an example of embedding. Sorry for the whiplash here. This might even be the plot that, or a version of the plot that you did, except that it's like active, and you can see that again. This is like you have these different cell types, and they're kind of sort of embedded. Like you can kind of like get pretty good classes. So you can imagine that a classifier in high dimensional space is probably going to be able to do a pretty good job with this. And that's indeed what we find. So like, so this code was the embedding, and then there's this analysis one. So to be clear, this was like after you have the embeddings, then what do you do? And so here, like this is SCGPT, and you can see like that, like a couple of interesting things to note is that there are like lots of different well isolated clusters, whereas this one is more maybe a little bit like the relationship, the discrimination between the classes is better in general here, but you can see that they have these like really horrible parts that are going to be completely impossible to differentiate. And then this one is, let's see, what's the difference between, well, it doesn't really matter. So like we do these, oh, this was the different versions of the embedding, and you can see like they're all kind of the same. This one gets a little better. Like I don't know, visually it's hard to tell, but like maybe this one and then this one, like slightly better probably, or actually this one. Yeah, so like this one's a little noisier, I would say, than this final one. So you can kind of tell visually that it seems to be doing a little better, but not dramatically, but better. So let's see, and then like this, I chose like this guy, and this guy. So TSP1, 2, and 14, and because they have good representation across all the cells, and if you kind of look, you'll see that basically there's not a lot of missing cell types, although they're like this cell type is only in 30, and so the classifier is probably going to not do well in 30, and maybe this type as well. What is that? Whatever that, that's this ectoepithelial cell, right? So, so then I basically just run like in a loop. I train, and I trained. Let's see, like if you look at what I did, it was sort of, leave one out, and then I'm looking at, let's see, I'm looking at the 3 different algorithms, which are random forest, XGBoost, or light GBM actually, and maybe one other. And then, so like blah, 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 lots and lots of compute. And then like, so here's some tabular data. And then eventually you get to this. And the other plot that you get to is maybe, well, okay. So I did this. I don't think the details you guys really care about a lot. But like, so, okay, first of all, this is how we ended up with this plot, is we embedded those stuff. Any questions about that? So this is partly about my workflow and stuff. So if you have like workflow questions or whatever, that's great too. And so, okay, how we did this, how did we do this? Well, again, no rocket science here, but if you look here, basically, we, sort of like for every gene, we submit batch requests to OpenAI. So the OpenAI embeddings are a little old. but they have a couple advantages. One is that they're Matryoshka embeddings. So, are you guys familiar with Matryoshka embeddings? Is it like a really good concept to know about?
I think
Okay,
there was one question in the chat about how are you quantifying which one is better, as in which metric. So I think about that last graphic.
Oh, okay,
No.
this one. Yeah, so this is the F1 score. So meaning, and this is actually an
Sorry,
insightful question. Yeah, go ahead.
I meant to ask about the when you were looking at the UMAP graphs, you were saying that some of the embeddings are better. So this is so you're quantifying it using the F1 score, the same models, right?
Yeah, yeah. And so the F1 score is a little bit like, it's not a great, the F1 score is only makes sense if you have all of the cell types in the same level in the hierarchy, right? So like way back in the beginning of the presentation here, I like you have this hierarchy. If you have like some cell types here and some cell types here, even if you're correct, you might get it quote unquote wrong. And so the F1 score doesn't work super well for like sort of broad data sets, but it works well for the data set I'm using to test here, because that Tabula sapiens data set, all of the cell types are at the same level in the hierarchy. Does that make sense?
Yeah. So for broad cell types, what kind of metrics do you use?
Sorry, what kind of?
Like when you are taking it from different families, what kind of metrics do you use other than F1?
So F1 precision recall.
No, I meant you said F1 is good for this particular data set.
Oh, I see. Okay, yeah. Let me. I'm going to get to that. So let me
Okay.
answer that in a little bit. That's a great question. And it's actually one of our most important questions. Yeah, I will answer that. Okay, so yes. So let's see. Yeah, okay. So, and I mean, like, there's no magic here. Most of this crowd probably knows how to do this. If you don't, I'll like go over it really briefly, but you basically, like in order to embed things with OpenAI, you basically, upload all your text to OpenAI in batch usually. And then so you create these files and then you just create a batch job, and it will, for something like, I think it's half the price or even less. It will just happily churn away at it for up to a day, depending on the size of the data set. I think you can see here and interestingly, for larger data sets, the throughput is much higher, but you can see like, for 30,000 descriptions, it took me about an hour? Not even an hour, half an hour or 40 minutes, right? So like, and it cost me like $1 or something. So like it's pretty amazingly cheap. Because their batch API is so well, like it's designed for like massive projects. So like it's super robust. Like you just send it stuff and you get back stuff in a little while, and it works well. And they've thought through all the failure conditions and how to restart and whatever. So like I was like really pleasantly surprised at how well it works versus trying to manage all that yourself. And it's half the price. Okay, so yeah, so that's like sort of how we generate it. And then, so we generate all these embeddings, we try it on different embedding types. And I was kind of thinking that, my hypothesis beforehand was that like actually adding extraneous or like less gene specific information and more about like the, diseases and pathways and drug interactions and whatever, that it would like not help or hurt the performance of the classifier, but it actually was wrong. My hypothesis was wrong. And it actually does, like, just adding more information about the gene just does tend to help. And it helped close the gap on the SCGPT. So, and then I've uploaded this all to Hugging Face, so you can, if you want to just use it for some reason, then you can just go and use it. Okay, and then, so that's like how we generated the embeddings. Okay, so good. Any questions? Oh, I was going to mention Matryoshka embeddings. this useful thing where 78 dimensions in the OpenAI large embeddings, but they were designed in such a way, and you can read the Matryoshka embeddings paper to understand the details, but they're designed in such a way that you can chop off the higher dimensions, and most of the information is contained in the lower dimensions, so that you can kind of adjust how much data you want to store and process versus how much you want to, what performance level you need. And even there are some cases when lower dimensions do better than higher number of dimensions. So we ended up using and I'll explain, we ended up chopping the dimensionality, but starting with the 78 dimensions. Okay, cool. So let's see, where are we? Sorry, one in the driveway. Yeah. Sorry, guys. Okay, so then the next thing that we did was we took, okay, so now we have this like sort of, we kind of believe this works. We have some embeddings we like. You know, we weren't very rigorous on this because one is it's very pragmatically focused on helping customers to get good results. So I mean, not that we shouldn't be doing evals, we absolutely have to be doing evals, but our focus is, okay, let's just learn about how these models work before we start doing really rigorous evals. So, and I think we've gotten to the point now, and I'll discuss where we need to start doing more rigorous evals. So here's, let's see, let's start here. So now let's load this Celix GNV2. This is 100 million cells. It's about, I want to say, I don't want to get this wrong, but I think it's like maybe 5 or 6 terabytes of data. So it's pretty big data. It's not massive, but it's not small either. And then, so one thing about this type of data is it's saved in this kind of interesting file format that it's like a file system in a file, and it allows you to store like the sparse gene expression data, but then also all the vectors of labels and other things, and then all sorts of metadata. And so that this is like the standard way that you represent this data, but what the consequence of this very flexible format is that the data's like first of all, there's a lot of cruft data that you, in any given circumstance, you don't really care about, or it's not cruft, but it's like irrelevant data. And then there's also means that different people put different like names on the same things and all sorts of, like, it's not very super well standardized. So that, you know, there's a lot of pre-processing you have to do. So I actually ended up writing a script to extract a bunch of, or like certain metadata form and write it to a certain metadata and write it to Parquet files so that I can do quick analysis on the metadata for the whole data set. So there's 1,000 or so files, and I wanted to be able to read the metadata and do statistics on them both so that we can curate a good training set. So I created the metadata in this file, and I can talk it there again. That's an open source project. It's right here. It's called ANData metadata. It's on GitHub. I can share that as well. And what it does is it creates you know, these Parquet files that you can use you can query it with DuckDB. You can load it as in Pandas or whatever your tool of choice is, so that basically we can, you know, sort of like ask, okay, what are the like genes being expressed in different files? What are the like number of genes? What are the, and then also I had to do some post-processing here on what are the distribution of gene counts. So like for a given file, it has this many genes expressed, and then like the number of active gene count, or sorry, the total number of genes, like gene expressions, and then the number of gene, like individual, or like the unique genes that were expressed. And so like I kind of plot this like in like a manual way with 3 files, and then go and get the distribution across, sorry, I'm gonna skip that, across all sorts of, sorry to scroll. Yeah, so then like, so now this is that data that I was just talking about, but this is every single, or not every single file, but many of the files in the data set, in order to understand, okay, like how similar are the data visually, right? So this is a thing I almost always do with large data sets, is I get like, let my eyeballs sort of understand the data and like find the outliers in the shape of the data. So like the things that I pull out from here is like, okay, these guys actually seem to be pretty uniform, but there's like some guys that are like small, like they have very small tight distributions, and others are wide, but they all kind of have a reasonable cutoff. These ones, like, these are really heavy tailed, and I cut them off as like this last column is rest. So the point I'm making is like, okay, I got familiar with the shape of data, and we were originally going to set some thresholds so that we throw away bad data, and we just realized from looking at this, we're going to train on everything. We're not going to any given file. We're not going to have any discriminatory factors that exclude data. Does that make sense? Okay, I want to make sure I'm not like boring you guys with sort of like data analysis details, but I wanted to like sort of talk about how we're selecting the training set. Any questions?
Yeah, it looks
Okay,
like Yax did ask if you had any intuition on why it ended up helping, but maybe in the interest of time, like you only have a couple minutes left. And I think it was a while back too. I just missed
yeah.
it.
Okay.
So in the interest of time,
Yeah.
we'll probably look forward.
Yikes, let me answer that on chat so I can finish up, but I will answer it. So okay, yeah. So what, like, from this crowd's perspective, the interesting thing about this data set and my training process was that or the data selection process was that these files are like this weird format that I mentioned, and I had to, like, so they're all in S3, and I had to use this like I had to rewrite the standard tools are just no good at using objects directly in S3. They want to download the whole file and then start analyzing it. And so that the upshot of that is that you end up, it's like a very prohibitive IO problem. And so that what I ended up doing was developing a couple tools, sort of in this process that were able that go and get file fragments. And then I ended up doing, and this is like where LLMs really shine, is I ended up doing this like integer linear program to select the best sort of fragments from the files based on their coverage of the cell types that I wanted. So there's like a whole bunch of work that I did. Like the vast majority of the training work that I did was actually just figuring out how to get the data set, the training set, out of the 100 million cells in such a way that it wasn't prohibitively expensive. Okay, so I got down to about 5 million cells, and then ended up sort of let's see. So I ended up building building the subset, and then I ended up we were going to use XGBoost for the classifier. XGBoost has problems because it doesn't scale well to large data sets that don't need a corresponding amount of processing, right? So you can scale it in various ways, but none of the ways it scales match this data set very well. And if people are interested, I can explain why. But so we end up using a multi-layer perceptron, and again, loading only the data, being very very selective about the data we load into memory. use Optuna to tune hyperparameters, and then we ended up with, you know, this classifier, you know, that is stored in weights and biases and can classify like our test set, you know, 100,000 cells in like 15 seconds. So like super efficient. At the end, the inference is super efficient, but it took a lot of work to get there. And we ended up with, so like, and let me just share on weights and biases really quick. In fact, I think it's referenced from here. So let me just grab it here. Yeah, here. So if you guys are not aware of weights and biases, you definitely should be using it. So like the like maybe the final thing I'll mention is that like I ended up, you know, learning a lot about training models in this process. I don't have a ton of experience with neural networks. And the interesting thing, so you can look at these two different training runs. One is the blue one and the, you know, the orange one or yellow one. And like I won't talk so much about my metrics. Actually, I want to talk also about the metric that we're developing. So I'll be really brief on this, but you can see that like this guy keeps having these loss spikes and never really getting to a point where it can converge before it has another loss spike. Whereas, so let me make this larger. So like, you know, it keeps getting down and then, oh shit, and then I have another loss spike, and this is a periodic thing. And what this corresponds to is different data sets. And so the reason is that it's doing this because the distribution in the data sets is different. And so that it like whenever it gets to a new data set, then it's like, oh crap, I'm mispredicting everything and I have to restart from here. And then it gets, you know, it like sort of like lowers so it sort of like gets down to a certain point and then doesn't have enough you know, generalization. So you can see it like generalizes a little bit, and that these loss spikes are a little bit not as bad, and it gets a little bit lower, but it doesn't really smoothly improve the neural network. So what we did was, this was using the 3078 dimensional embeddings. And what we did was, okay, just chop it off so everything can fit in memory, so we can easily shuffle around the data set, and then just shuffle the entire data set and pick random subsamples for my batches, and then suddenly the loss is dramatically better. And you know, these are different measures that are not super important, but one. So I'll stay on for a few minutes after and answer questions, but I wanted to answer the question about the metric. So we developed the metric that we use. Let's see, it's written out somewhere here. Yeah, here. The metric that we used, so you can, if you look at the macro metrics, so like the standard F1 score macro, then they're total garbage. Like the F1 score here is 9%, right? And precision and recall are equally bad, right? So like and the reason is because even if I'm correctly predicting, but I'm in the wrong place in the hierarchy, then I still am mispredicting. And so like these, you know, so these are ranking metrics that are traditionally used in information retrieval. Like this is like is my correct answer in the top 10, top 5, top 2, and like these guys are just basically weighting like how far from the top are my is the correct answer. And so so those do better, but I find it very unsatisfying because like one, if you like in practice, I'm not going to be like, that doesn't help me, right? Like I'm doing science, I don't want to know like it's one of these 5. I want to know what's my best guess, right? And the other thing is that you know, it doesn't take into account this hierarchy very well. Where did that slide go? It doesn't take this hierarchy into it well. So I think that we're coming on like basically a hierarchical metric that we want to use that is basically how far in this hierarchy am I from the correct measurement. So, okay, this is kind of biology related, not so probably interested for this crowd, but I do like, I thought that this was really fascinating because this is very much a process of discovery of the problem. I'm learning about the problem as I learn about and the data as I learn about the specific model. So I thought the process here was really exciting to me, and I wanted to share it with you guys. Did I answer the woman who was
Yeah,
asking the question?
yeah, I got it. Yeah.
Okay, good. All right. And
Yeah.
I'm curious, have you ever dealt with something like this? And is that it like, and if so, like, what did you do?
I haven't worked with bioinformatics data at all. So yeah.
Okay. But what about like hierarchical data in general?
Not really. No, I worked with like EEG related data, and all of those are. I don't see any hierarchical kind of connections in
Okay.
that.
Yeah.
I also
Yeah.
wanted to ask, like, how important do you think having domain knowledge is in economics is to get into this field, like to do this bioML stuff?
I think that having an expert to ask questions and guide the decision-making process is pretty critical, right? 'Cause like the intuition for what will work, what's important, those things, you need an expert, but I don't think you need to have a formal training. I don't, right? So I have a friend who is a very experienced bioinformatics who's done drug development and other, sort of related stuff his whole career. So so that we work together on this, and I bring the AI, he brings the bio, and like we end up, we're working really well together.
Yeah. And do you have any suggestions for like quick exploratory projects following up on this, on your work right now?
I mean, like I'd love to, like work together on something. I can definitely come up with
Okay.
specific stuff. I mean, yeah, why don't just DM me and we can talk about ways that you could help. I mean, I could think about an answer if what we're doing is not super appealing, but you want something else like in the same domain or whatever, I can also probably come up with something for that.
Okay, thank you.
Yeah.
Message.
Awesome. Yeah, okay, good. So I guess that's probably all I have time for. Do you guys want to, are there any other questions? I'm happy to stay after and talk. But I want to like sort of, at least have an informal ending there. Or a formal ending, I guess. Okay, okay, cool. Well, thank you guys for joining. I hope that was helpful and interesting. Please, if you have questions or follow-ups or need links or whatever, just hit me in Discord and I will share what you need and answer questions, and would love to collaborate and all that. Okay, take care, guys. And I guess Yikes or Flo or whomever, we usually, we're starting to publish these on the Latent Space TV, right?
I have a bunch of recordings that I'm overdue to publish. So yes, they will be published at some point, but if you need it sooner, just let me know and I'll upload it.
I mean, I like to, after I do a talk, I like to just, I like to get as much credit for all the work I do as possible. So I like to post it on X and LinkedIn,
Yeah,
just to say,
I can send you the raw if you want, and then once it's on YouTube, I'll send you the link too.
okay. I mean, I think I probably and honestly won't do anything with the raw. So
Okay.
like as soon as you can get the YouTube up, that would be awesome.
Yeah, no, I'll do that today. Sounds good.
Okay. Okay, cool. Thanks. Awesome. All right, guys, thanks. Take care.
You
Bye.
do-